Okay, very good afternoon, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagitial. In the CPGET 2024 pre-online coaching, my dear young aspirants, I wish you all the best. Yesterday is the last date for your application, online application for CPGET 2024. So today onwards, the payment on the penalty base, you those who are not yet applied, so hurry up and apply your CPGET 2024. And also listen carefully. Today onwards, our sessions is also definitely increase. Daily 2 to 3 sessions will going to take the classes every day. So in this regard, today our eminent resource person, Dr.P.Manjula Madam, Assistant Professor, Department of Botany, Food and Nutrition, Rbvrr Women's College Autonomous. Madam, please welcome and share your screen. Start your session, Madam. Please welcome.
Okay, thank you, sir. So today's topic is vascular cambium. Okay, good afternoon, one and all. And thank you, sir, for giving this opportunity. So today we'll discuss about the next, in previous classes, we discussed about the internal structure of root, stem, and shoot apex and root apex, and theories regarding root apex and shoot apex. So today's class, we will discuss about the vascular cambium and secondary growth. So vascular cambium, generally it is a...
if possible, please click on slideshow, Madam, please.
Okay. So next topic, that is the vascular cambium. So vascular cambium means, so it is a type of meristem, that is a lateral meristem. So generally it is present in between the xylem and phloem, that is present in the vascular tissue, that is in between the xylem and phloem, and mainly helpful in the formation of secondary growth. Okay, so the vascular cambium, that is plural is cambia, or simple, that is cambium. So it is a lateral meristem in the vascular tissue of the plants. So generally this cambium, which is present in the vascular tissues of the plants, mainly in the dicots. So the vascular cambium, it is a cylindrical layer of the cambium that runs throughout the stem of the plant that undergoes the secondary growth. So here when we observe the structure, so here this one. So, so in between the xylem and phloem. So this red color or orange color one, that is the xylem, and here yellow color one, that is a phloem. So in between this, so here the cambium is present. So this thin layer, so it is nothing but the cambium. So it is only meristematic during the secondary growth only. Okay, so during the secondary growth, it becomes meristematic and undergoes cell division to produce the secondary xylem towards inside and secondary phloem towards the outside. So this one, vascular cambium. So here this one is a xylem, and so this one is a xylem and this one is a phloem. So in between this, so this thin layer, it is the cambium. So if the cambium is present between the xylem and phloem, so it is called as the open type of vascular bundles. So location of the vascular cambium. So generally in dicots, so it is present in between the xylem and phloem. So the vascular cambium is in the dicot stems and roots, which is located between the xylem and phloem in the stem and root of the vascular plant. And it is the source of both the secondary xylem growth and secondary phloem growth. So generally from this vascular cambium, that is the secondary growth will takes place. Okay, it produces secondary xylem towards inwards and secondary phloem towards outwards. So here, this one is the cambium, vascular cambium. So this one is a vascular cambium. So it produces xylem towards inward and phloem towards the outward. So here the bark and here the carp cambium. So another cambium, that is a carp cambium, is also present. So it produces the bark. So in monocots, that is in monocot stems such as the corn, palms and bamboos, they do not have the vascular cambium. So generally in monocots, that is the cambium is absent. Hence the vascular tissues also, closed type of vascular bundles are seen in the monocots. So hence here they do not exhibit the secondary growth by the production of concentric annual rings. So they cannot increase in width by adding the lateral layers of the cells as in conifers and woody dicots. So here this is the internal structure of the dicot and monocot stem. So here in the dicot stem, that is the cambium is present between the xylem and phloem. But here these vascular bundles, they are scattered in the cortex only. Okay. So here the cambium is absent in monocot stem. So next one is the types of vascular cambium. So here again based on the position, so here the two types of vascular cambium are present. That is the intravascular cambium and intervascular cambium. So intravascular cambium means the cambium that arises within the bundle of the primary vascular tissue of the stem. It is called as the intra. That is the cambium which is present in between this xylem and phloem. It is called as the intravascular cambium. So intervascular cambium, so it is a strip of intravascular cambium, generally which becomes jointed by the additional strips of the cambium, which are called the intervascular cambium. Okay, so during the growth, here the formation of this intra and intervascular cambium united to form the cambial ring. Next one is the organization of the vascular cambium. So generally, the cells of this vascular cambium are different from the general meristematic cells. So here the cambial cells, they are vacuolated. So what are the characters mean? They are the vacuolated, and the resting cells have the less vacuoles. But the active cambial cells, they have the highly vacuolated cells. Okay, so the cambial cells are of two kinds. That is, it is made up of two initials. That is ray initials and fusiform initials. So ray initials. So these are much smaller than the fusiform initials, and they are slightly elongated and nearly isodiametric in shape. And they produce the radial system of the cambial zone. And another initial, that is a fusiform initials. So the shape is spindle shaped. So a fusiform initial, however, it is prismatic cell in its middle and wedge shaped at the ends. So here this one is the ray initials, and here these are the fusiform initials. So ray initials, these are the ray initials, mainly helps in the radial growth, whereas the fusiform initials, they helps in the horizontal growth. Okay. So next one is the next one is the structure and function. The intravascular and intervascular cambium join up to form the ring. So intravascular means the cambium which is present. For example, here this one is a vascular bundle, that is a vascular bundle. So here this one is the xylem and this one is the phloem. So this is the intravascular cambium. So like that, so during the secondary growth, what happens? So these all are the vascular bundles. Okay. So during the secondary growth, so these are the cambium. So here xylem. And here this one is a phloem. So in between these, so this one is the intravascular. So during the growth, secondary growth, this intravascular and the cells which are present here, the parenchymatic cells are present. So the cells which are present, they becomes meristematic and act as the intervascular cambium. So here they form the ring-like structure. Like this, so they form the ring. It is called as a cambial ring. So this one is the intra and inter in between. So this vascular bundle, so they forms a cambial ring, and they produce the xylem towards inside, secondary xylem towards inside, and secondary phloem towards outside. So the intravascular and intervascular cambium join to form the ring here. So the separates the primary xylem and primary phloem by this cambial ring. So this cambium, they separating the cambial ring, separating the xylem, primary xylem and primary phloem. So the vascular cambium produces the secondary xylem on inside of the ring and secondary phloem on the outside. So here this cambium, it undergoes. So here the cambium, it is nothing but it is a lateral meristem. So it becomes only active during the secondary growth only. Okay. So by taking the signals and due to the hormones, so they becomes active and they undergo cell division to produce the secondary xylem towards inside and secondary phloem towards the outside. So the vascular, they produce the secondary xylem on the inside of the ring and the secondary phloem on the outside. So the pushing the primary xylem and phloem apart. So what happens? So why the new cells are coming? So the early formed cells becomes pushed or crushed. So maintenance of the cambial meristem. So the vascular cambium is maintained by a network of interfacing signal feedback loops. So currently both hormones and short peptides have been identified. So generally for this growth, so here the hormones are also involved and some peptides also involved during the secondary growth. So similar regulations which occur in other meristems of the plant. So the cambial meristem, which receives the signals from the both xylem and phloem sides of the meristem. So here during the secondary growth, this hormonal regulation also plays an important role, that is during the second cell division. So the hormones that are involved in the vascular cambial activity are auxins, ethylene, gibberellins, cytokinins, abscisic acid, and mainly. So they are involved in this cambial activity, and each one of these plant hormones are vital for the regulation of the cambial activity and are dependent on their concentration. So next one is a cell division in the vascular cambium. So generally here this cambium they produce the secondary xylem and phloem. So how these divisions will occur means, so when the cambial initial which produce the secondary xylem and secondary phloem, they divide periclinally. So periclinal divisions means that is parallel to the cell here, cambium cells. So it is periclinal division. Anticlinal means that is a perpendicular to. So at one time, a derivative cell which is formed towards the xylem, at the another time towards the phloem side. So one time they produce the cells towards xylem side, inside, and sometimes they produce towards the outside, that is the phloem. So it is not necessarily in alternation. So each cambial initial which forms the radial files of the cells, one towards the outside and other towards the inside. And the two files meet at the cambial initial. So like this here. So this one is the cambium. So it produces the xylem one side. So and another time, so it producing the phloem. So here this cambium which producing the xylem, secondary xylem towards inside and secondary phloem towards the outside. So these divisions are periclinal divisions. So parallel to this cambium. So this one is the initial cell. So they produce the phloem mother cell and here the xylem mother cell. So it produces the xylem and phloem. So next one is the seasonal activity of these vascular cambium. So in plants which growing in the tropical regions, this cambium is active throughout the life, and in contrast to this. So generally in some plants, that is the this cambium is active and they produce the secondary growth. But in contrast to this, in plants of temperate regions, so this cambium almost ceases its activity with the commencement of the unfavorable seasons. So generally in temperate regions, what happens? This cambium only active during the favorable seasons only, and they produce the secondary growth. So generally in atom are in this dormant stage, which remains till the following spring. So when it is reactivated and produce the new xylem and phloem. So during the unfavorable condition, this cambium becomes inactive. It stops its activity, and during the favorable conditions, so they becomes reactivated and they produce the new xylem and phloem. So these cambial cells which take up water and enlarges radially and divide periclinally. So this vascular cambium and cork cambium, these are the secondary meristems which are formed in the stem and root. So we already discussed about this vascular cambium. So this vascular cambium mainly helps in the secondary growth, that is production of secondary xylem and secondary phloem. Whereas the cork, so after the tissue of the primary plant body have differentiated, so this vascular cambium is responsible for increasing the diameter. So what happens due to the increasing the cells? So the diameter of the stems also increases. and forming the woody tissue. So the cork cambium. So the cork cambium which produces bark. So this is present towards the cortex region. And so what happens? So the epidermis will ruptures due to the increase in these tissues and they produce the bark. Okay, this cork cambium produces the bark, produces some bark. Whereas a vascular cambium, they produce the secondary xylem and secondary phloem. So here the vascular cambium, so this red color red ring, so it produces the secondary phloem towards outside and secondary xylem towards inside. So this one is the central medulla, primary xylem, and here this one is the secondary xylem, and here cambial ring, and here this one is our secondary phloem, and this one is the primary phloem. So here cork cambium. So they produce the outer layer, that is bark. So this so periderm. So it is this layer it is called as the periderm. Cork cambium, they produce the periderm. So the vascular cambium and secondary growth. So in dicot stems, the vascular cambium initially differentiates from the procambial cells within the vascular bundles. So in monocots, they do not have the vascular cambium. And some of them, such as the palms and other Joshua artery, which exhibits a secondary. So sometimes, even though the cambium is absent, but they show secondary growth. So they have a thickening meristems that produce a secondary ground tissue. So here the cambium is absent, but the meristem, thickening meristem, they produce the secondary ground tissue. Okay, so increase in the width of the stem and additional vascular bundles, which differentiate the secondary ground tissue. So here the secondary growth is absent, but due to the production of this secondary ground tissue, they become thick in monocots. So next one is another case, that is the abnormal secondary growth. So it is a abnormal type of secondary growth. So that occurs in some arborescent monocots. So generally in monocots, cambium is absent, but here anomalous secondary growth will take place. Some exceptional cases like Dracaena, Yucca, and agave. And storage roots also, that is in the beetroot and sweet potato. So in arborescent monocot stems, the secondary cambium, which grows in hypodermal region, and the later forms the conjunctive tissue and the patches of meristematic cells. So these meristematic patches grow into secondary vascular bundles. So here they produce the secondary growth and conjunctive tissue. So we will discuss this again in the next topic. So anomalous vascular bundles, they also occur in the cortex, that is called as the cortical bundles. So the cells which are present in the cortex region, they becomes active and they involved in this production of this secondary tissue. So it is called as